Immunology Insights, Basics and Beyond, Chapter 17, Paul Ehrlich and the Side Chain Theory. Marcello here. Welcome back to Immunology Insights, Basics and Beyond. In our previous chapter, we explored the major histocompatibility complex MHC, a group of molecules that are central to the adaptive immune system's ability to recognize and respond to pathogens. Today, however, we will turn our attention to one of the most influential figures in the history of immunology, Paul Ehrlich. His groundbreaking side chain theory revolutionized our understanding of how the immune system recognizes and responds to pathogens, laying the foundation for the modern field of immunology. So let's talk science. To introduce Paul Ehrlich, Paul Ehrlich was born in 1854 in what is now known as Poland. From a young age, Ehrlich showed a keen interest in science, particularly in the burgeoning field of chemistry. He pursued his medical studies in Germany, where he became fascinated with staining techniques used to identify different types of cells under the microscope. This interest would eventually lead him to make significant contributions to the field of hematology and immunology. Ehrlich's curiosity and innovative spirit drove him to explore the mechanisms by which the immune system recognized and responded to foreign substances, leading to the development of his famous side chain theory. Let's now have a closer look at his famous side chain theory. At the turn of the 20th century, the scientific community was still grappling with the mysteries of how the immune system functioned. The prevailing theory was that the immune response was a somewhat non-specific process, with little understanding of how antibodies worked at a molecular level. Ehrlich thought to change this with his side chain theory, which he proposed in 1897. Ehrlich's side chain theory was inspired by his studies on how cells bind to specific dyes. He hypothesized that cells have side chains, which we now know as receptors on their surfaces that can bind to specific molecules, much like a lock and key. So, when a foreign molecule or antigen binds to one of these side chains, it triggers the cell to produce more of these side chains, which then are released into the bloodstream as antibodies. His side chain theory included three major aspects. First, specificity. Each side chain is specific to a particular antigen. This specificity allows the immune system to target and neutralize pathogens with great precision. Second, antibody production. The binding of an antigen to a side chain triggers the cell to produce and release large quantities of that side chain into the bloodstream, which we now understand as the production of antibodies by B cells. And third, memory. Ehrlich also suggested that the cells could remember the antigen, leading to a more rapid and robust production of antibodies upon subsequent exposures. This concept laid the groundwork for our understanding of immunological memory. Although the side chain theory was initially met with skepticism, it quickly gained acceptance as it provided a plausible explanation for the specificity and adaptability of the immune response. Ehrlich's theory was a precursor to the clonal selection theory, which later expanded on his ideas and became the foundation of modern immunology. Ehrlich's side chain theory was a significant breakthrough in immunology and had a great impact. It provided the first detailed explanation of how the immune system could specifically recognize and respond to an almost infinite variety of antigens. This concept of specificity became a cornerstone of immunology, influencing countless studies and leading to the development of vaccines, diagnostic tests, and therapies. Ehrlich's work also contributed to the understanding of autoimmunity. He was one of the first scientists to propose that the immune system could sometimes mistakenly target the body's own tissues, leading to autoimmune diseases. This idea was initially controversial, but has since become a fundamental concept in the study of immunology and autoimmune disorders. Paul Ehrlich left a great legacy. He was not only a brilliant scientist, but also a deeply passionate and dedicated researcher. His work habits were intense. He often worked long hours, driven by his insatiable curiosity and desire to understand the mysteries of the immune system. Ehrlich was known for his despite the different approaches, Metchnikoff focusing on phagocytosis and cellular immunity, and Ehrlich on antibodies and humoral immunity, the two men respected each other's work and even shared the Nobel Prize in Physiology or Medicine in 1908. Ehrlich's legacy extends far beyond his scientific contributions. He was a pioneer in the field of chemotherapy, coining the term magic bullet to describe a drug that could specifically target a pathogen without harming the host. This concept led to the development of the first effective treatment for syphilis, salvarsan, which was discovered in Ehrlich's laboratory. Today, Paul Ehrlich is remembered as one of the founding fathers of immunology. His side chain theory laid the foundation for our understanding of antibody production and specificity, and his work continues to influence the field of immunology and beyond. To conclude, Paul Ehrlich's side chain theory was a monumental achievement in the history of immunology. It provided a framework for understanding how the immune system specifically recognizes and responds to pathogens, paving the way for modern immunological research and therapy development. In our next chapter, we will explore the already mentioned inflammatory response more in detail, a critical component of the immune system's defense against infection and injury. Stay with us as we continue to uncover the fascinating complexities of the immune system. Before closing, I have a small request. If you enjoyed my content and want to hear more, like and subscribe. It's a small step for you, but it helps me a lot. So, see you next time and thank you for listening.